North Carolina Agricultural and Technical State University was established on March 9, 1891 as the first land-grant institution in North Carolina. As an HBCU, NCAT currently is the top producer of degrees awarded to African American students in North Carolina and top 3 in research universities within the UNC system. Specifically within the Department of Natural Resources and Environmental Design, the department provides students the skills and knowledge to explore issues surrounding food production and air, water, and soil quality in order to tackle real environmental issues our country may face. This project is many of the invigorating research projects available on campus through the Jakai Lab. In the ever-growing need to understand and improve plant production versus known pesticides, graduate student Felicitas and Ewunsoh observed the involvement of glucosinolates in the feeding habits of the Harlequin bug in different varieties of stages of mustard and collard plants. The Harlequin bug, or the Magenta historica, is one of the most important insect pests of the cabbage mustard in the southern part of the United States. By sucking out the sap of the plant, the bug causes the plant to wilt, brown, and eventually die. The life of the cycle of the Harlequin bug lasts 50 to 80 days, with the insect going through the egg, nymph, and adult stages. Harlequin eggs are typically laid in batches of 12 and can be distinguished by their black and white stripes. They take 29 days to hatch into nymphs, and from there they live as nymphs for 4 to 9 weeks before reaching mating age. After this, the adults go from their nymph form to the shape we most commonly know as a stink bug with its flat shield-shaped body. Like many pungent plants, like mustard, cabbage, etc., contain glucosinolates, which are a glucose-derived sulfur-nitrogen secondary metabolite found within the leaves. The metabolite is produced by the mustard oils within the plant that are released when chewed or damaged. Due to its bitter taste, it naturally acts as a defense mechanism against plants and diseases. However, for pests like the harlequin bug, the presence of glucosinolates can allow for the insect to identify its next host. So following leaf production, what was first conducted was the dual-choice arena test. This test is used to determine the feeding preference of the adult harlequin bugs between collard, Georgia Southern, and the mustard varieties at different growth stages. For the materials, we had 4 petri dishes, paper towels separated into 4 quads, scissors, early vegetation at 35 days, late vegetation at 45 days, flowering vegetation, adult harlequin bugs, water, a 38mm metal cork borer, and a camel brush. For the procedure, a paper towel was cut into a 15 cm diameter circle and placed into a 15 cm by 2 cm depth petri dish. From here, the paper towel was split into quadrants. Using the cork borer, leaf discs of 38mm diameter were cut from the leaf samples collected and placed in opposing quadrants with 2 collard leaves and 2 mustard varieties. From here, 4 10-day adult harlequin bugs were introduced to the center of the petri dish using a soft camel brush. And the data collected was collected every 6 hours, 3 times a day using the following damage scale. This was then repeated for the remaining mustard growing stages. The next test was using the olfactometer to determine the likely role of semi-chemicals in mediating the observed preference of harlequin bugs and crucifiers. The materials include a 4-arm olfactometer, a purified air supply system, and 4 2-gram samples of the mustard leaf variety. For this test, 2 grams of the leaf variety are placed in the arms of the machine, and the air supply is allowed to run for 5 minutes to stabilize the release of volatiles or odor from the leaves within the main chamber. From there, the 4 adult harlequin bugs are introduced to the center of the chamber and allowed to roam for an hour. The placement of the insects within the arms are recorded every 15 minutes before the insects are returned to their chambers. This is repeated 4 times to account for every growing stage. The next tool used is the spectroradiometer. Using the plants grown within the field in the greenhouse, this allows for the plant's glucosinolate production to be tracked and measured. After setting a reference for the sensor, 10 measurements were taken per variety from 1 leaf on each plant. The meter is then hooked to the sensor and computer, which produce a graph showing wavelength absorption. By using the absorbance collected, it's possible to estimate the amount of glucosinolate within the leaves. So for the results, within the dual-choice arena test, it was found that the old-fashioned or OFM varieties were favored by the harlequin bugs. With this, the leaves were separated, dipped in nitro, crushed, and frozen to be sent to a collaborating lab